Pluripotent stem cells induced from mouse somatic cells by small molecule compounds. Let's find the professor and learn more about this, cause I don't have a clue on this stuff.
Woo! What are they talking? I think it's a new experiment. Let's find out!
Professor, can you explain induced pluripotent cells and this new alternative method to make them?
Sure, let me explain that to you. Well, pluripotent stem cells, such as embryonic stem cells, can self-renew and differentiate into any cell type found in the body. Somatic cells can be reprogrammed to become pluripotent. The two usual procedures to induce pluripotency are nuclear transfer and the use of exogenous pluripotency-associated factors. But an alternative method for inducing this pluripotency consists in using small molecules. They enable the reprogramming in the absence of OCT4, which is one of the reprogramming-associated factors normally used, and it's involved in the self-renew of undifferentiated embryonic stem cells. These small molecules have some advantages because they can be cell-permeable, non-immunogenic, more cost-effective, and also more easily synthesized and preserved.
Professor, you have kind of a girly voice, don't you? What about the methods? How is this done?
Okay, the cells used as a starting point in the search for the small molecules were mouse embryonic fibroblasts expressing 3 out of the 4 pluripotency factors normally used, except OCT4. They were expressing OCT4 promoter-driven GFPs, which allowed them to be identified in the colonies that were able to express this gene even without the 4th factor. This could mean that the combination of molecules used could replace the gene's function. VC6T was the first molecule combination used in the treatment, and it only enables reprogramming of somatic cells in the presence of OCT4. We found out that using VC6T plus a molecule named FSK turned some cells colonies GFP positive, which meant that this molecule properly replaced the missing factor, inducing reprogramming pathways.
Oh god, this seems very complicated, but what is the next step?
Well, the next step treated the GFP-positive colonies with VC6TFZ, a combination of the previous molecules plus DZNEP, a molecule that inhibits DNA methylation, making the genes more accessible. The result of this combination so far is a higher ratio of GFP-positive colonies after DZNEP addition. The last step is to obtain the chemically induced pluripotency stem cells was based on switching the medium to a dual inhibition medium, which ended up giving the GFP-positive colonies an embryonic stem cell-like morphology.
So, the next step is characterization. The chemically induced pluripotent stem cells obtained were compared to embryonic stem cells in some important parameters like gene expression profile and DNA methylation. Results show that the gene expression profiles were similar in chemically induced pluripotent stem cells and embryonic stem cells. Both express some of the most important pluripotency markers like NANOG and OCT4 via immunofluorescence and RT-PCR.
Professor, your voice is changing a little bit. Are you okay? And what are those pink dots?
Yeah, I'm fine, whatever. The pink dots are the cell colonies expressing the different pluripotency markers. Using bisulfite sequencing, the DNA methylation state at OCT4 and NANOG promoters in the chemically induced pluripotent stem cells were identified to be similar to that in embryonic stem cells, which gives good reliability to this method. And the second figure is the RT-PCR result.
Okay, Professor, so tell me more about those pluripotency factors that you said before.
So, starting the in vivo part of the experiment, the CIPSCs obtained were injected into skid mice. They were able to differentiate into respiration, neural, and pigmented epithelia and muscles, tissues from all the 3 germ layers. And unlike chimeric mice generated from transcription factors, the mice generated from iPSCs were 100% viable and apparently healthy for up to 6 months.
Woohoo! 6 months? That's good news.
Okay, Professor, but what about those essential molecules? How did you find out which molecules are we using exactly?
To determine which molecules were actually essential in this reprogramming process, each one of them was individually removed from the cocktail given to the cells. The molecules removed that generated the least amount of GFP positive colonies were the ones we considered essential to this combination.
Okay, tell me more about the gene profile of the cells.
The final step of the experiment aimed to analyze more deeply the genes related to the pluripotency in the CIS species. The most relevant genes were SAL4, SOX2, GATA4, GATA6, SOX17, and OCT4. In the first scenario, it was demonstrated that SOX2 and SAL4 were responsible for stimulating OCT4. As OCT4 and SOX2 are considered master genes due to their relevance, they activated and repressed other pluripotency genes on the pathway of reprogramming cells. Their most important outcomes are the activation of NANOG and suppression of GATA6. It's known that NANOG is a transcription regulator that imposes pluripotency and prevents ESC differentiation, and GATA6 plays a role in vertebrate organogenesis. Thus, what these genes basically do is maintain the CIPSCs in the pluripotent state, which is what we're looking for. Yay!
Okay, I think I got it. So, the conclusion is that this study presents an alternative and reliable method to induce pluripotency without using genetic manipulation, using exogenous factors that can be difficult to manipulate. Those small molecules modulate molecular pathways related to pluripotency within the cell itself, besides being more cost-effective and more easily synthesized and standardized. These molecules' effects on inhibiting and activating specific proteins can be finely tuned by varying their concentrations. All this combined with the fact that CIPSCs maintain normal karyotype makes these cells valid for further investigation to apply them in therapeutic approaches in the future.
And most important, we don't die anymore.
Woo!
No, wait! We just live a little longer. Oh man, so there's no escape.